Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, I have a great guest for you all. Her name is Dr. Karen Sugden. We're going to learn all about assessing the reproducibility and integrity of epigenetics in this episode. We're going to define reliability and what it means, how this actually impacts epigenetic research on a large scale, how we can deal with the unreliability and fix this for better outcomes and better insights as it relates to the research. We're going to talk about how we measure methylation using beadchips and what that process looks like. And we're also going to talk about two of Dr. Sugden's amazing studies, one looking at blood-based DNA methylation with age-related cognitive impairment and dementia, and a lifetime marijuana use study looking at that relationship between marijuana and epigenetic age. A quick introduction to my guest. Karen Sugden is a scientist from Duke University in the Department of Psychology and Neuroscience. As part of the Moffitt and Caspi research team, her current research focus is on the collection and analysis of biological information in large longitudinal cohorts, most notably the Dunedin longitudinal study and e-risk study, which you all are very familiar with because of my past podcast episodes. She started out working in human clinical pathology before changing track to genetics, graduating from the University of Liverpool in the UK. And she then joined the Social Genetic and Developmental Psychiatry Center at King's College London, where in 2004 she received a PhD in the study of genetic and genomic influences on behavior and mental health. It is here that Karen was first fortunate enough to work with the professors, Dr. Moffitt and Dr. Caspi, on the Dunedin study, a relationship that still continues to this day. I hope you all enjoy this one. It is essential to talk about the reliability, integrity, reproducibility when examining these epigenetic DNA methylation markers. And now for my guest, Dr. Karen Sugden. Welcome to the Everything Epigenetics podcast, Dr. Sugden. Super excited to have you here today. Thanks for joining.
Oh, it's great to be here. Thank you. That's very enthusiastic and I like it.
Well, you know, we haven't stopped giggling since we just started and chatted beforehand, but you know, this is the first time we've actually ever had a conversation online. I'm very familiar with your work, but would love to start off by just learning more about you. So, you know, where did you get started, your career, and you know, where you are up until this point?
Um, I guess we can start right at the very beginning and a long time ago. I've always kind of been interested in genetics, so that's kind of my field of study. I was interested in genetics even as a little kid. My parents wouldn't like to know that had a breeding program for gerbils in my bedroom, and I had recordings of all the color combinations that I was getting. So I didn't even know I liked genetics as a child, but apparently I did. But actually, I then, when I left school, actually started working in a pathology lab, which we were doing histology and cytology, which was very, very interesting. But, you know, even then, we kind of had a little bit of genetics involved in that, and I kind of got even more interested in it. And I decided that it's time to go and take this a bit more seriously. So I embarked on a degree in genetics. And it's whilst I was doing that I actually got interested in genetics and behavior. It was actually to do with animals at that point, but you know, I mean, I thought it was so cool that genetics could be related to behavior. And once I've got my degree, I wanted to take that further. And actually, I was interested in humans because humans are a lot more complicated than animals, and I thought that would be cool. So I moved to London to the Social Genetic and Developmental Psychiatry Centre, which is part of King's College London, to start PhD genetics of mental health and human behavior. And that's where I started working with Terry Moffat and Avsham Caspi. And they introduced me to Need and Study at that point. First, I'd heard of it, but I was just blown away by it. So we did some work together, and that kind of never stopped. And I'm still doing work with them now. That's been going on now for a couple of decades, which is very, very cool. As part of that, we actually ended up moving to the US. So we moved from London to Duke University in the US. And a couple of things have actually happened since then. I've always been kind of a lab-based person, but the way that research goes has moved more towards using bioinformatics style tools. So our work has moved more towards genome-wide types of approaches and doing more bioinformatic work. So I do much less work in a lab these days. But also, as part of working with the Dunedin study, we've sort of watched the members get older. I've been with them for quite a long time. So now we've sort of moved our focus more towards aging as we're watching them age, as we all do. But so that's a couple of things that's kind of like happened in my work in these last few years.
Beautiful, beautiful background. I love the story of, you in your room when you were a young girl and, kind of studying this phenomenon, even though you didn't really know what it is. So it does seem like that interest was there at a very young age. So just super interesting. And then obviously all the work that you've done in conjunction with Dr. Caspi and Moffat on that Dunedin cohort and study is absolutely incredible. I love to see how these, insights and kind of the questions we're asking changes over time as maybe the cohorts do as well. So I appreciate you giving that background. Now, a lot of your work and definitely what we're going to focus on today, I haven't discussed this with anyone. That's why I was excited to have you on, is going to be on patterns of reliability. And I think that's obviously very, very important in this epigenetic methylation world or, DNA methylation space. So can you just talk about, what is reliability and why does it matter in the scope of what we're focusing on?
Sure, of course. So reliability is just simply the extent to which something can be measured more than once and to actually give the same answer when you do it a second time. So, this applies to any measure. It applies to anything across all branches of science, engineering, medicine. This is not an epigenetic specific thing. I mean, it even applies to you measuring out stuff when you're baking a cake. If you can't use your measuring cup and give you the same answer, your cake's not going to turn out the same way. So, reliability is really important for everyone. So, to give sort of a very silly example of what it might actually be, imagine you're going to measure the height of someone. You grab your tape measure, you stand them up against a wall, you measure them, they're 5'5". Great, okay, they're 5'5". And you ask them to go and stand up against the wall again. This time they're 5'6". What does that mean? Are they 5'5"? Are they 5'6"? So you can't rely on that measure. It's not reliable. You get out a second tape measure, you do it again, they're 5'5", both times. You've got a lot more confidence that that person's 5'5". That second tape measure is giving you a lot more validity in their measure. So this is really what, in a nutshell, reliability is. It's just consistency across repeated measurements. So why does this matter? Well, it matters because it's fundamental to research. If you can't get the same value when you measure something twice, you can't be sure of any findings, any conclusions, or anything that you make using that measure are valid. So if we went back to our height example, again, silly example, but imagine you didn't know your tape measure was unreliable. You didn't know that it wasn't giving you the right measures. And then you went and you used it to measure a bunch of people. You got everybody's heights. And it turns out you're interested in what these people like to eat as a snack, right? And then when you look at everybody's preference, it turns out everybody over 5'6", well, they like chips, and everybody under 5'6", they like nuts. Great. Okay. You take this to a marketer. And marketer says, great. We don't have to spend money trying to sell our chips to short people. They like nuts. So, I mean, this sounds like it's perfect, but you don't know that your measure is unreliable. It turns out you've got a bunch of short people in the tall group. You've got a bunch of tall people in the short group. And you know what? That relationship between preference and height just isn't there. And so this is, if you extend this and apply this to scientific research, you can see now why this is a problem. I mean, that's a silly example, but this really is a problem. If we make discoveries based on measures that just aren't reliable for whatever reason, it might just be random error or whatever, but it means our conclusions aren't valid and we're not able to actually replicate these findings. So, this is not something we want if, for example, we're trying to find treatments for disease, or we've got drug interventions or trials or things like that. Because, say if we were trialing a drug that was based on an incorrect or unreliable measure, then we're going to put people's lives at risk. We can harm people. Plus, on the other side, science is really, really expensive. It's really expensive, and if we're going to spend time and money trying to replicate things that were never really there in the first place, this is not a good way to spend our money and our time. But also, I mean, on the flip side as well, we might end up missing all this important stuff because we can't find the relationships that actually really are there. We might miss, to take the drug example again, if we have an unreliable measurement testing a drug, we might miss the fact that actually there is a treatment option there, and we might just throw it out into the trash. So, I mean, reliability is really, really important.
Yeah, seems like if you don't have that part of reliability, and I love the example with the height and the chips and nuts. I think that's good. It's a waste of time and resources, right? And it's almost like you're kind of maybe going down those rabbit holes, which are the wrong rabbit holes, or maybe not the correct ones, in which you actually think there may be a specific finding. So I just think this is super prevalent. And you had your specific paper, I think it was around April 2020, titled "Patterns of Reliability: Assessing the Reproducibility and Integrity of DNA Methylation Measurement." So that's the one paper I was like, yes, I need to talk to her about this. So yeah, relating it, I guess, more to epigenetics, how does this unreliability factor arise in epigenetics?
Mm-hmm. Well, I think the best way to kind of explain it is to perhaps go right back to basics, and think about how we measure epigenetics. Um, so, the most popular and the most accessible way that we can assess epigenetics is looking at DNA methylation. And I'm sure all your viewers will be more than familiar with DNA methylation. But the way we commonly do it is we use something called microarrays. And they're just little glass slides, but they have thousands of tiny little short DNA sequences. Actually, I have one. I have one here. See if I can get it up on the screen. Here's one. Oh. Yeah. So I just have one lying around. So there you go, a treat. So they're just these little glass slides, and they just have like thousands of these really tiny short DNA sequences stuck to them. They sit on beads and they like wave their little tails in the air, just looking for pieces of DNA that might be floating past. So each one of these little tiny strings tags a region in the genome that you're interested in. So when we were interested in DNA methylation, we're actually interested in regions of the genome that we know can be differentially methylated. So there's really two things about this little DNA sequence. We call them probes, because they basically probe the DNA sequence that's flying past them. It's complementary to the DNA that we're interested in, because DNA really likes to stick to its opposite. So when it sees its opposite, it will grab it. So it's kind of, it's probing for that opposite bit. And also that there's two versions on these arrays. So we've got a bit that will specifically grab methylated bits, and we've got a probe that will specifically grab the unmethylated bits. One thing where we can find that unreliability might come up is that some probes are actually a bit better at being selective than others. So some probes might not do quite as good a job as grabbing what they should. They might grab slightly too much of the wrong stuff, So you're coming up. This is what we know, technical error. So I mean, this happens. So this could be one way in which reliability might pop up. What we also have to remember is that when we are sampling our DNA, whether it's from blood, saliva, or whatever, we're sampling. We're taking thousands, if not millions, of cells, and each of this cell has got its own copy of the genome inside. Where for any particular point of the genome we're interested in, it's either going to be methylated or unmethylated. So when we're measuring DNA methylation, we're actually measuring the proportion of methylated genome copies in all the DNA samples that we've got within our particular sample that we've put on the array. And that can range from 0 to 100% methylated or unmethylated if you wanted to do it the other way, but we usually talk in terms of methylated. So as I mentioned, I'm bringing this up because it's just worth remembering that some probes are just a bit better at picking out the right bit than others. Some are really good and some aren't so good. And that's why I wanted to just go over the background a little bit. Well, also, I just wanted to give a brief background about the microarrays that we doing, because this is actually pertinent to what we'll be talking about in a bit. So they contain hundreds of thousands of probes, each one measuring different parts of the genome. And the most popular ones are the ones that I actually just held up for you. Those are made by a company called Illumina. And their microarrays have gone through a series of improvements over time where they content has changed slightly, or beadchips as they actually call them. And they've actually expanded them and changed them ever so slightly in every iteration. So the first one started out having 28,000 probes, to probe 28,000 regions of the genome. And then research developed and we learned more about DNA methylation, and we learned there were more regions that were interesting. So they expanded this to 450,000 probe sites. And that was known as the 450K array. And more recently, they've actually developed it again. We've now got 850-ish thousand probes, and we call that the EPIC bead chip. So at each iteration of this, the older chip was kind of like had new content put with it, and it got bigger, and they took out some of the probes that don't work quite so well that they knew about, so that each bead chip is essentially a bigger version of its previous one. But also another thing to bear in mind with these microarrays is that they're actually generic. That even though they're human specific, they're generic in so much as they're designed to work for all different types of human tissue. So they're not the arrays we use. We generally deal in blood DNA. Most people do. It's a very easily accessible tissue, so people like to use blood. It gives you lots and lots of DNA. It's great. However, these arrays that we use don't just target the profile that you might expect from a blood DNA sample. They also have to include targets that you might expect from other tissues, which won't always look the same as blood DNA, because each tissue will have its own specificity. It'll have its own pattern. That's part of the way that tissue gets regulated, so that tissue can become what it's meant to be instead of becoming something it doesn't. So, they have to be generic. So what it means is that when we look at these microarrays and all the data that we get on it, not every single probe is going to be relevant to our study, because if we've got blood, we won't need to worry about probes that aren't supposed to be different in blood, but they are supposed to be different, let's say kidney or something like that. So that was a long discussion of how things can actually become unreliable, in so much as, setup of what the arrays look like and how probes work. So
yeah, that's great. I don't think anyone has described like that entire, methylation bead chip kind of array-based technology from Illumina. So that's great. I do have kind of just one question, if anyone else is wondering it too. So why don't you think, maybe it's not Illumina, maybe it's some other company builds, those separate arrays based on tissue type? Is it just a cost factor? Probably. I mean, obviously that's not cheap or easy to do.
yeah, I mean, it's to be perfectly honest, you'd have to ask Illumina's board for that, whether they would be worth doing or any other company or whatever. I think there are application specific arrays. Yeah, so I think some possibly exist. I think it's just because it's also a field where we can't find things out until we look for them. We kind of want to have everything because you don't know until you look, right? So if we try and cut it down, we might miss something because we didn't know when we made the array that that's actually really is important. It's just we haven't spotted it before. So in some ways, it makes sense to have as comprehensive a platform as you can, just in case there is stuff hidden away down but you hadn't decided to look before. So does that answer your question?
Yeah, no, that's a great point. I mean, that makes, we don't know what we don't know, right? And then, of course, Illumina's coming out. I don't know what they're calling it. I think it's the Epic V2, right? I think it's going to be like the next step up where it's like 1.2 million positions somewhere. Yeah. So that'll be exciting. But again, we're getting more access, right? Because there's like 28 million different methylation markers in every single cell type. So we're creeping up on that number slowly, but hopefully.
Right, correct.
So yeah, no, that was a great answer. And yeah, I don't know if you want to go into more of your paper or you want to talk a little bit about how we deal with these unreliable probes. I know it's kind of all interworked together. So yeah, would love to hear your thoughts there.
Yeah. So, I mean, I think in a way I might just talk about the background why we decided to look at this because um, on the surface it might not seem that exciting, but it was integral to our whole research program. So, yeah. So what actually happened is this paper started as just kind of like an exercise for our team on what we do with data, how we process data, how we deal with data. Um, so just as a bit of background, we were about to collect DNA methylation data from the Dunedin study participants at age 45. Now, I think we've talked about Dunedin study previously and with other um, people who've joined you in this. But briefly, it's a longitudinal study. Everybody born in a town in New Zealand, called Dunedin, 1972-73. They've been followed up progressively all throughout their life. Well, when they were 26 and 38 years old, we were able to collect DNA and we profiled their DNA methylation from DNA that we collected those two ages. We did that using the 450K array, the 450K bead chip that we talked about earlier. So when we came to do the same again at age 45, well, guess what? The 450K bead chip didn't exist anymore. It had been superseded by the Epic. So we sat down and we said, okay, so are we going to be okay combining all our data together? They're different bead chips. We don't know. Are we going to be able to do this? Because one of our key aims is to have longitudinal data that we can use over time, but we can't use longitudinal data over time if it's not the same data over time, because you kind of need to make sure that you're not introducing error. So we said, okay, let's just go to literature, let's go and find out whether we're all right to do this. And we went and looked, and there's a little bit there, but not so much, telling us that there is some similarity between the two, but some of it isn't, some of it is. And we were like, well, okay, so what does that mean for us? Because it's all right documenting what this similarity is, but if it has consequences for the kind of research we want to do with these data, then we've got issues, and we couldn't really find anything that told us their answer. So we said, we'll just test it ourselves. Let's see what happens. So that's where this paper came from. Came from us wanting to know whether we can use the data in the way that we want to use the data. So we were actually quite lucky because we had some bead chip data, some 450K and EPIC bead chip data from twins that are part of another one of our studies, the ERIS study. And so we had 350 individuals that we've measured twice. So basically, we had a reliability study ready to go where we could test the reliability of the 450K array data versus the EPIC array data. So we got those data, we subset it to just the probes that are shared between those two arrays, and that ended up being about 440,000-ish, I think. And then we just asked, well, how similar are they across the arrays? In other words, how reliably could the methylation level be determined across these arrays? And actually, we found on average that probe values were pretty unreliable. And in fact, only about 7% of probes, when we looked at them, could be deemed to have excellent reliability. I mean, it's all a scale, but we have a sort of a standard scale on what we deem excellent. So that, out of the 440,000, it's not that many. So, I mean, that sounds concerning, but it's actually what we expected, to be honest. And the reason for that was that we already knew that some probes were going to work better than others. I mean, it's just a fact of any molecular assay. You're going to have error that creeps in. Some things just don't work as well in the chemical environment that you're subjecting to them to as other ones do. It's just par for the course. So we knew that that's always going to happen. We also knew that our arrays weren't specific to our blood samples. So we're going to have a bunch of probes on there that shouldn't be delivering us any data. We think, we don't know, but but we also knew that a couple of other people had reported in the literature that actually this is what happens, and it looked very similar to the results we were getting. So we weren't surprised, but it still didn't answer why. Why should we bother? What are the consequences? Should we even bother? I mean, it may not be a problem. Who knows?
Right. Yeah. That does sound super concerning. But like you said, you were expecting that. And now I think I have more questions for Illumina than anyone else. But, that's kind of what we're going through right now with the 850K Epic V1 to the V2. It's like, hey, here are the, kind of sites that are going to be missing. And some of those sites are included in algorithms, which we use, which is super concerning. So, it all makes sense. And yeah, I'm just curious as to, what the cutoff is, why certain probes are maybe let out, if they're doing, I'm sure they're doing, internal studies and kind of looking at the comparison. But like you said, considering that the tissue type may be different and then you're excluding some probes altogether, that accounts for probably a large amount as well. So yeah, super interesting. Do you think you're going to do the same thing with the Epic V1 versus V2 or try another type of reliability study there?
I think from this point onwards, given what we found with in this paper, where we found that reliability really does impact the work that we do, it can have big ramifications. We will always, going forwards, always perform a reliability test on any new DNA methylation data that we generate for that reason. And that's not to denigrate Illumina's products. Then the Illumina's products are great. It's just that we want the answer for ourselves.
Yeah, I think you have to, though, to make sure that your data is reliable. So you can, especially with the Dunedin cohort, how you've studied it over, 50 plus years, right? You have to make sure that you're able to compare that data. So no, I think that's great. And I think that's something that this field, the researchers are super interested in learning more about and understanding. So what do you do? What do you do with that information, Dr. Sugden? What's next? How do you take that and say, okay, we need to, translate this in, this way or whatnot?
So, I mean, there is no set answer to this. So, I can give advice to people when they contact me and ask me what to do about this. I can tell them what my preference is. I mean, it may be right or it may be wrong, depending on on who you are. But I mean, if the kinds of values that we have generated in terms of reliability in this study are they're very similar to those that other people have generated doing similar studies. They're not identical, but because each study is going to have its own technical variation that impacts measurements ever so slightly, but they're very similar. So one thing that people could do is they could take all these values that we've generated and just say, okay, I'm going to assume that probe number 1 has a reliability value of 0.8 because that's what it says on this list, and go forth with that, and then decide to perhaps just use the probes that meet a certain threshold of whatever they deem to be good reliability. ideally, what people might want to be doing, and this only really is going to work for people who are generating data, is to actually factor in replicability, factor in repeated measures as part of their study design. So instead of just measuring the, say, like 200 samples that you've got, measure 220, and then you've got for 20 samples, you've got a pair that you can test reliability on. So basically just do exactly what we did and just take these samples and say how similar are the two values when you run this twice. Then you would have a reliability list that's specific for your sample.
Yeah. I mean, that makes sense. The latter makes sense. It's just unfortunately a little bit more expensive, but if you had unlimited funds, you might as well. The first point you made about saying, you have all these probes, you look at the reliability, you assign a certain, weight to that probe and then make a threshold. This is included, this is not included. Are you talking more about like the pre-processing type of analysis, maybe in terms of kind of giving those probes different weights?
Not necessarily. I mean, if you so if you imagine we've got a list of, say, we've got 440,000-ish that we have reliability values assigned to, that we've determined reliability for, essentially. um, you could literally take that list, say, I've got a data set, I've got 200 individuals that I measured on the EPIC or a 450K array. I'm going to cross match probes with this list, and then I'm going to filter out anything that doesn't reach a threshold that I predetermine, whether that's, 0.5, 0.8, whatever. I mean, it is up to individual researchers to decide what's appropriate and what's not. So then, you might cut that down to 120,000 probes instead of 400 and something thousand probes, because you might kick out a bunch of unreliable stuff. So that's one way to do it. So that actually would be the easiest way for a researcher to do it, to be perfectly honest, because it doesn't really require any sort of further statistical manipulation of your analysis. Um, yeah, yeah.
That makes sense. And this might be the wrong question to ask as well, but those probes, you said they're grabbing a particular part of the genome, right? Like a specific CPG. Is that correct to say? So what if these probes, as you're going through them, right, you say, oh, this one's unreliable, but there's, 10 that are in the Dunedin pace algorithm, right? So how do you account for that?
Well, the short answer to that is, when it comes to Dunedin pace, we only have reliable probes in the algorithm, because one of the consequences of this work that we were doing, and as I say, this came before Dunedin pace development, of which I was a member of the team that has done that. One of our prerequisites, that after we discovered that, unreliability does have some consequences for a number of different facets of research that we're interested in, we decided that we would only use reliable probes that we deemed reliable based on our data when we were developing the algorithm. So even if we applied this filter to our data, we shouldn't kick out any of our Dunedin pace probes, because they were never in the algorithm in the first place.
And I'm going to add that to the list of reasons why I love the Dunedin pace algorithm so much, is you don't even have to deal with that. No, that's great, Dr. Sugden. And then, in terms of who's at fault here, there's, no one's really at fault, right? This is just part of using a generic array technology. And, guess what? It's going to be the same for every type of technology. You're going to run into some type of unreliability, but then create different pathways to, uh, to be able to improve and again, go through those kind of processes for that particular technology. So, and in terms of it being the same for every chip or B chip, I'll say, I'm going to guess it's going to be different, right? The unreliability, or what if you're going, from 850K chip to another 850K chip? How are the probes compared there?
So the short answer is, who knows until we test it. I wish I could be a bit more concrete than that. All I do know is that for the data that we had, the 450 versus the EPIC, for the 350 twins that we looked at, these are what the reliabilities came out to look like. Is it going to be the same for every comparison that you do? Probably not. It's going to be very, very similar because we've seen that against other people's work. However, it's not going to be identical. And the only way we're going to find out is actually run those experiments. So, when we have the opportunity to do the comparisons, we'll do it and we'll find out the answer at that point.
Sure. Absolutely. Do you have anything else to add? I know we talked about this a little bit earlier too. Do you have any other reasons as to how unreliability can impact research? Is there anything else you want to add there?
So, I think epigenetic data is getting used a lot. It's really popular. We have just acres and tons of data available for public use. It's great. It's fabulous. This is one of the goals of generating these data. Public availability, get people to use them. What this does mean is that not everybody's going to be familiar with the actual data itself, the nitty gritty, how it was made, what it looks like, or even be familiar with like all these factors that we're talking about today. They're not going to be familiar that unreliability exists. They're not going to be familiar that all the probes aren't necessarily made equal. They're not going to be familiar with all those things. So I think I'm probably going off a little bit off topic now, but I think one of the things that we want to do is we want to try and make sure that this is one of the things that becomes aware to people who aren't familiar with these data, because what happens is people start using them and start reporting things, and they're not aware that it's not set in stone what this value is, because they're not aware that some unreliability might be connected to it. I think one example where this is actually kind of highlighted quite well is actually in the paper, and we were interested about association studies, which is like bread and butter for these large cohorts. People love to do association studies. They're great. They give you some really interesting insights. So kind of like briefly, all we're doing when we do an association study is we're testing for the strength of a relationship between an exposure and an outcome, so maybe the exposure or the outcome, in this case, DNA methylation.
Mm-hmm.
So you then see whether that reaches any sort of predetermined statistical threshold for significance. And if that strength of association between DNA methylation probe and your outcome is strong, then you're like, oh, it's associated, great. So when we do these kinds of tests using epigenetic data of the type that we're talking about here, we're doing thousands of association tests. We're doing like 400,000 or 850,000 of these tests. And we're looking to see which of those 850 or 450,000 probes are actually past this threshold. So what we wanted to know, because they're super popular to run, you see them everywhere, we wanted to know whether this unreliability had any consequences for this kind of research. So we decided to look at cigarette smoking. And the reason for that is it's one of the most strongly replicated, it's a robust finding when it comes to DNA methylation. Cigarette smoking really does mess up your methylome. So, it's one of those things that you can kind of rely on finding something with. So we went into literature and we found all these genome-wide association tests where they've taken all these bead array data and looked at cigarette smoking and looked which probes were associated. We found at that time there were about 22, I think it was. And then we took all the probes that they'd reported as being significantly associated with smoking. And all we did is we just counted how many times that probe popped up in a list across all those 22 studies. And it was kind of like a sort of a lazy way to check replicability of a probe. It's like if a probe is replicable, it'll keep coming up time and time again. So you should have a high number for that count. So, what we found is that when we did that, those probes that had a high number, those that kept coming up time and time again, those were the most reliable probes from our definition. So these are the ones that you could measure reliably time and time again. We could also find regularly when we look for the same thing. And then those probes that didn't really pop up very often, those were the least reliable. So, this is like a huge impact, because if people are reporting findings from association studies, but they're actually finding results with unreliable probes, they could potentially be false positives, and it sends people, like you say, down rabbit holes. It sends people looking for things that can't replicate because they were never really there in the first place.
Yeah, yeah. And epigenetics, biological age. I mean, it's thrown in your face every single day. It's this hot, sexy topic. And very trendy right now. But I always tell people it's a trend that needs to stay. We need to measure reliable information over, a longitudinal analysis to know what's really being affected. And I think what we need to go back to, and exactly why I'm having this conversation with you today, is about education, right? Like I can't stress enough how little I think we even know about the methylome alone, let alone people doing all of these studies. We just really need to kind of have the community come together and work on this and talk about these different issues. So no, I mean that makes perfect sense. And I think as I'm going through now reading literature and interviewing people, I'm going to have this in the back of my mind, kind of looking to see if they mention reliability or have, a paragraph about that in their methods or, whatever section of the paper. So no, this is great, Dr. Sugden. Now, you did mention, the two ways that we can actually deal with that unreliability. Anything else you can think of, or again, like going through papers and making sure we're cross-checking?
Yeah, I mean, the repeated measures on the actual data, especially, I think is just like it's the most important thing. We're in a huge sort of data production period. We're in a huge data production era. We're being encouraged by grant funding agencies to, to create these data, share them, get them used, and that's great because they're expensive, but they're valuable. Use them. Um, what would be great is if we also had some sort of prerequisite for doing some sort of this reliability checking on all these data. And don't get me wrong, people are, I mean, I'm not trying to suggest nobody's doing this. People do this. But if we made it standard, we wouldn't have to write these papers because, it would already be inherent in everything. So I think that is the gold standard. That is the gold standard, to have data with these metrics that is specific for that particular sample set. We understand. I mean, it is expensive. It is time consuming, and it's difficult to do when you do longitudinal measures because you kind of have to do it every time you collect new data, which gets very time consuming and expensive. It's it's important, but it is a time suck. So. That would be the gold standard. Yeah. But I think, sorry, go ahead. No, I mean, in lieu of that, all I was going to say, in lieu of that, just some recognition, I think, that this exists. I think this is one of the hardest things, is getting the message out, because it's kind of like people who've worked with these data for a long time already inherently know it. They may not have tested it, but they know that this is a thing. I mean, it's not, nobody talks about it because everybody knows it. But then the people who don't use these data from, raw up to research probably aren't as familiar with this being a concept. So, they go into this naively, thinking that a probe is a probe is a probe, and it's not necessarily the case. So it would be, one of the easiest ways to get around this would be for a request that whenever somebody publishes maybe an association study, for example, with specific DNA methylation probes, journal authors, so journal editors suggest that they put the reliability metric right next to the probe, so that we can say, oh, do we believe this or not? It might just cut down on some of that, like, chasing that people have to do.
No, I love that. I think you're exactly right. It should be standard, and then you're wondering if the people who are editing these journals know about the reliability, right? So I think it's just, again, about education, the people who are already in the field, hopefully educating those who are, just now entering the field and maybe trying to learn a little bit more. So this has been a beautiful conversation. I'm so excited that we, brought light to it. I'm going to switch gears completely. I have a couple studies I want to ask you about that you've published and you've worked on. So we've been talking about the Dunedin longitudinal study. Huge fans of everything that you've done for that. But you do have a paper titled "Association of the Pace of Aging Measured by Blood-Based DNA Methylation with Age-Related Cognitive Impairment and Dementia," and I can picture the graph from this paper. I know exactly what it looks like and what this paper tells us. So can you describe what you did and what you found here?
Sure. So in this paper, we were interested in assessing the extent of association between the epigenetic clocks. Again, I'm sure your viewers are more than familiar with epigenetic clocks. In this case, we were interested in looking at the Horvath clock, the Hannum clock, the pheno age, the Grimm age, and Dunedin pace. So we were interested in what they look like when we associated them with various measures of cognitive impairment. So, like asking them screening tests that people take when they are being assessed for dementia, and cognitive tests where you, for instance, have to recall a certain number of words after a certain length of time. And this is in a bunch of older people that it's not actually a Dunedin study at all, but it's in a cohort called ADNI, which is the Alzheimer's Disease Neuroimaging Initiative. This is a huge study that's been going for a number of years, monitoring their older age people, and they're interested in watching them as they move towards having a greater risk for dementia. So we were fortunate. We got access to this resource. So there's all these cognitive measures and diagnoses of dementia, cognitive impairment, and things like that. But they also have DNA methylation data. So that's how we were able to calculate these clocks for the individuals in this. And we really wanted to answer one specific question, which is faster aging related to greater cognitive impairment? Because this is something that we really needed to answer. People will want to know this. We want to know, can we take measures of faster aging, some of which are developed in younger people, and apply them to a group of older people who are experiencing cognitive impairment? So short answer.
Mm-hmm.
So no matter what test we looked at, whether we looked at whether we looked at a clinical diagnosis of dementia or mild cognitive impairment, whether we looked at those screening tests, or whether we looked at those cognitive tests where, like the word recall and things like that, those who were aging the fastest, as measured by Dunedin pace specifically, these were the guys who were scoring the poorest in all those domains. So why is this important? Well, what it does mean is that we can use information from epigenetic pace of aging measures like Dunedin pace, and track cognitive impairment, potentially before it even starts. And this is one of the goals of intervention work, is to have a tool that you can use before people are already in the last stages of what you're trying to intervene on. So this is why this was actually very exciting, that we can measure things in older people related to cognitive impairment.
Sure. Yeah, I think that's fascinating. Yeah, I have an APO34 variant, very interested in Alzheimer's. My grandma had dementia, Alzheimer's definitely runs in the family. So I think it's something, people are very familiar with and they can definitely relate to it as well. So I just had to ask you about that one because as I was going through your papers, that one caught my attention. And for those listeners who are like, show me everything, I want to read everything, I'll link everything and all of the studies. That way they can go ahead and look more into your work. Another study, haven't talked about this yet on the show, but more recently you published one. This is titled "Lifetime Marijuana Use and Epigenetic Age Acceleration, a 17-year prospective examination." So tell us about this one, Dr. Sugden. Haven't talked about marijuana on this show quite yet, so I think people would be interested here.
Sure. Well, this paper is, we published this, my wonderful colleagues at the University of Virginia. They're great. And it kind of seems a bit timely. You know, marijuana's been in the news quite a bit recently, so kind of interesting that the timing on this one. But what we actually did for this is we were looking at the effect of marijuana use.
Mhm.
This was Grim Age, and actually the precursors to Dunedin Paste, Dunedin POAM, because you know we've been at this work for quite a bit. And we were looking in a group of middle-aged individuals that Joe Allen, who is the first author on this paper, has been tracking for an awful long time. So we know a lot about them. Now, there was a number of really interesting things that we found in this paper. The first thing is marijuana use, lifetime marijuana use, increases epigenetic aging. So those people who are using marijuana are aging faster than those who aren't using it so much, which, you know, that's an interesting point, number one. And that's actually true even when you take into account a person's own cigarette smoking. So if you kind of set everybody's cigarette smoking value to 0, you still see that association. So it is kind of not smoking driving this association, because oftentimes smoking will be associated with faster aging, as I think everybody, common sense will tell you it probably is. So second, we found that there's this dose response relationship between faster aging and marijuana use. So the more you use, the faster you're aging. So you know, you can see this dose response moves up over time. The third thing that was interesting was that the fastest aging was observed in those people who'd used it most recently. So it's like this proximal effect, so that, you know, you aging faster as you've just used it, as opposed to people who haven't used it for ages. Who, I don't know what that means, whether it's some sort of recovery effect. We can't speculate on that from these data, but that's an interesting observation. The fourth observation was, and I think very interesting, that if you take into account the DNA methylation profile of one particular probe, when we're looking at this relationship between faster aging and marijuana use, it kind of, if you make that, set that as 0 for across everybody, so make everybody equal for that, this relationship actually breaks down, which I think is fascinating, because it kind of suggests, now this particular probe, it's one of the most replicated probes associated with cigarette smoking, tobacco smoking. It's actually the one that would replicate 22 times in our reliability study, because whenever anybody looks at tobacco smoking, they find this probe. And biologically, it's related to hydrocarbons, you know, that would be inhaled normally in smoking. So it kind of suggests, this finding kind of suggests that some of that relationship between faster aging and marijuana use is being mediated via some sort of inhalation of perhaps hydrocarbons or something else. Now, what was really interesting was this effect was even seen in people who didn't smoke, said they'd never touched a cigarette. So this isn't anything to do with them perhaps smoking cigarettes and marijuana, because even people who don't smoke cigarettes were seeing this. So it's kind of interesting suggestion. It's only a suggestion that perhaps this effect between faster aging and marijuana might have something to do with the inhalation. So kind of interesting. It threw up a few interesting avenues for future research, I think.
Yeah, definitely. I don't know if like secondhand smoke or like anything would maybe have to do with that, but yeah, I know there's a lot of questions and a lot of studies done in obviously smoking and epigenetic methylation markers. So I appreciate you giving a review of those two papers there. We're almost at the end here. A couple more questions for you, Dr. Sugden. What are you currently studying? What's your time being spent on currently?
Well, at the moment, currently, we have a paper in review that is looking at the link between education and genetics and pace of aging, and we're super excited about it. Right, so this is really love this paper. So the premise here is that we know there's a link. It's long been known there's a link between education and health. You know, people who get more education generally have better health outcomes, and people who don't, you know, if you follow them or even cross-sectionally, they just seem to have better health. There's also a pretty substantial amount of literature around the genetics of educational attainment, and in particular, polygenic scores of education. Now, a polygenic score, I don't know, you may or may not have heard of this term, but it's essentially where you look at a person's genome, you take all the parts that have been associated with, well, in this case, it would be education, and then you just add them all up. Okay, so it's just like a sum of how much education-related genetics you have in your genome, and this is at a genetic variation level, not an epigenetic variation level. So then each person will get their own score based on how many education-associated genetic variants they actually have. And these scores do predict how much education a person is likely to get. This has caused some concern for some people because it sounds a little bit like genetic determination, but it's a very active field of study. Now, however, in this paper, what we were interested in, what we wanted to know was, does this positive effect of education on health, so in this paper, it's specifically looking at the pace of aging as measured by Dunedin pace, does this persist even against this backdrop of this education polygenic score? So in other words, can people with genetic risk for lower education still benefit from getting an education, get those benefits of education that lead to slower aging? And the answer is yes. And that's really great news. So the message is education is good for all, in spite of your genetics. Doesn't matter. Well, doesn't matter for
Right.
but education is good, which I think is a fairly positive message.
I think so too. I think that's great to end on, and yeah, I will make sure to look out for that study. So yeah, and just keeping up with all of the great work that you're doing. This next question is one that's a curveball. I always say I don't put it on the agenda, but Dr. Sugden, if you could be any animal in the world, what would you be and why?
Oh my goodness. So I don't think I've met an animal that I don't like yet, which is slightly a problem for me and for anyone else associated with me. But I think it would be so cool to be a tardigrade, the little water bears. Yeah, they're little bugs that float around, but they're indestructible.
Indestructible bug. That's what you want to be.
Oh no, that doesn't come across very well, does it?
I like it. I like it. You're strong. You're water. Are they the ones that walk on water?
No, I think they still float. They float around. I know that when they were in one of the Marvel movies, they were in there with Ant-Man, I think. I have not, no. There you go. As long as there's tardigrades in it, I'll be fine.
There you go. Well, we've come to the end of this amazing podcast. For listeners who want to connect or see your work, where can they find you?
Email is a great way. So just my name, karen.sugden@duke.edu. Drop me a line over anything. I will try and get back to you as soon as I can. Other than that, you can go to the Moffitt-Caspi website, which is moffittcaspi.trinity.duke.edu, I believe, or just Google Moffitt-Caspi, you'll find them, no problem. And you'll find not only me, but all my lovely team members that I work closely with, and all an overview of all our research that we do as a team.
Definitely. Yeah. I'll put that out there for everyone to view. And yeah, you know what? Thank you so much, everyone, for joining and listening here at Everything Epigenetics podcast. Remember, you have control over your epigenetics. So tune in next time to learn more. Thanks so much, Dr. Sugden.
Thank you.